Entangled Life, Chapter 2, Living Labyrinths, Part 3. Mycelium usually grows from hyphal tips, but not always. When hyphae felt together to make mushrooms, they rapidly inflate with water, which they must absorb from their surroundings. This is the reason why mushrooms tend to appear after rain. Mushroom growth can generate an explosive force. When a stinkhorn mushroom crunches through an asphalt road, it produces enough force to lift an object weighing 130 kilograms. In a popular fungal guidebook published in the 1860s, Mordecai Cook reported that, quote, "Some years ago, in the English town of Basingstoke, a road was paved, and not many months afterward, the pavement was observed to exhibit an unevenness which could not readily be accounted for. In a short time after, the mystery was explained, for some of the heaviest stones were completely lifted out of their beds by the growth of large toadstools beneath them. One of the stones measured 22 inches by 21 and weighed 83 pounds. If I think about mycelial growth for more than a minute, my mind starts to stretch." In the mid-1980s, the American musicologist Louis Sarno recorded the music of the Aka people living in the forests of Central African Republic. One of these recordings is called Women Gathering Mushrooms. As they wander around collecting mushrooms, their steps tracing the underground form of a mycelial network, the women sing amid the sounds of the animals in the forest. Each woman sings a different melody. Each voice tells a different musical story. Many melodies intertwine without ceasing to be many. Voices flow around other voices, twisting into and beside one another. Women gathering mushrooms is an example of musical polyphony. Polyphony is singing more than one part or telling more than one story at the same time. Unlike the harmonies in a barbershop quartet, the voices of the women never weld into a united front. No voice surrenders its individual identity, nor does any one voice steal the show. There is no front woman, no soloist, no leader. If the recording was played to 10 people and they were all asked to sing the tune back, each would sing something different. Mycelium is polyphony in bodily form. Each of the woman's voices is a hyphal tip, exploring a soundscape for itself. Although each is free to wander, their wanderings can't be seen as separate from the others. There is no main voice. There is no lead tune. There is no central planning. Nonetheless, a form emerges. Whenever I listen to women gathering mushrooms, my ears find their way into the music by choosing a single voice and riding with it, as if I were in the forest and could walk up to one of the women and stand next to her. To follow more than one line at a time is hard. It's like trying to listen to many conversations at once without flickering from one to another. Several streams of consciousness have to commingle in the mind. My attention has to become less focused and more disturbed. I fail every time, but when I soften my hearing, something else happens. The many songs coalesce to make one song that doesn't exist in any one of the voices alone. It is an emergent song that I can't find by unraveling the music into its separate strands. Mycelium is what happens when fungal hyphae, streams of embodiment rather than streams of consciousness, commingle. However, as Alan Rayner, a mycologist specializing in mycelial development, reminded me, quote, "Mycelium is not just amorphous cotton wool," unquote. Hyphae can come together to form elaborate structures. When you look at mushrooms, you're looking at fruit. Imagine bunches of grapes growing out of the ground in their place. Then imagine the vine that produced them, twisting and branching below the surface of the soil. Grapes and woody grapevines are made of different types of cells. Cut up a mushroom and you'll see that it is made of the same type of cell as mycelium, hyphae. Mushrooms grow into other structures besides mushrooms. Hyphae grow into other structures besides mushrooms. Many species of fungus form hollow cables of hyphae known as cords or rhizomorphs. These range from slim filaments to strands several millimeters thick that can stretch for hundreds of meters. Given that individual hyphae are tubes, not threads, it is easy to forget about the fluid-filled space within the hyphae. Cords and rhizomorphs are large pipes formed from many small tubes. They can conduct a flow thousands of times faster than through individual hyphae. nearly 1.5 meters per hour in one report, and allow mycelial networks to transport nutrients and water over large distances. Olson told me about a forest in Sweden where he had observed a large armillaria network that fruited over an area the size of 2 football fields. A small footbridge crossed a stream that flowed through the area. I started looking more closely at the bridge, he remembered, and saw that the fungus had started to wind its cords under the bridge. It was actually crossing the stream using the bridge. How fungi coordinate the growth of these structures remains a mystery. Cords and rhizomorphs are good reminders that mycelial networks are transport networks. Body's mycelial roadmap is another good illustration. Mushroom growth is another. To push their way through asphalt, mushrooms must inflate with water. For this to happen, water must travel rapidly through the network from one place to another and flow into developing mushrooms in a carefully directed pulse. Over short distances, substances can be transported through mycelial networks on a network of microtubules, dynamic filaments of protein that behave like a cross between scaffolding and escalators. Transport using microtubule motors is energetically costly, however, and over larger distances, the contents of hyphae travels on a river of cellular fluid. Both approaches allow rapid transport across mycelial networks. Efficient transports allow different parts of a mycelial network to engage in different activities. When the English country house Haddon Hall was being renovated, a fruiting body of the dry rot fungus, Serpula, was found in a disused stone oven. Its mycelial connections wound back through 8 meters of stonework to a rotting floor elsewhere in the building. The floor was where it fed, the oven was where it fruited. The best way to appreciate flow within mycelium is to watch its contents shuttle around the network. In 2013, a group of researchers at the University of California at Los Angeles treated mycelium so that they could visualize cellular structures moving within the hyphae. Their videos show hordes of nuclei surging along. In some hyphae, they travel faster than in others. In some, they travel in different directions. Sometimes traffic jams form and nuclear traffic is rerouted on hyphal slip channels. Streams of nuclei merge with each other. Rhythmic pulses of nuclei, nuclear comets, rush along, branching at junctions and darting down side ducts. It's a scene of nuclear anarchy, as one researcher, Riley, observed. Flow helps to explain how traffic circulates within a mycelial network, but it can't explain why fungi might grow in one direction rather than another. Hyphae are sensitive to stimuli, and at any one moment are confronted with a world of possibilities. Rather than extending in a straight line at a constant rate, hyphae steer themselves towards appealing prospects and away from unappealing ones. How? In the 1950s, the Nobel Prize-winning biophysicist Max Delbruck became interested in sensory behavior. He chose as his model organisms of fungus Phycomyces bleiklianus. Delbruck was fascinated by Phycomyces' remarkable perceptual abilities. Its fruiting structures, essentially giant vertical hyphae, have a sensitivity to light similar to that of the human eye, and adapt to bright or low light as our eyes do. They can detect light at levels as low as that provided by a single star, and only become dazzled when exposed to full sunlight on a bright day. To provoke a response in a plant, one would have to expose it to light levels hundreds of times higher. At the end of his career, Delbruck wrote that he was still convinced that Phycomyces was the most intelligent of the simpler multicellular organisms. Besides its exquisite sensitivity to touch, Phycomyces preferentially grows into wind at speeds as low as 1 centimeter per second, or 0.036 kilometers per hour. Phycomyces is able to detect the presence of nearby objects, a phenomenon known as the avoidance response. Despite decades of painstaking investigation, the avoidance response remains an enigma. Objects within a few millimeters cause the fruiting body of Phycomyces to bend away without ever making contact. Regardless of the object, opaque or transparent, smooth or rough, Phycomyces starts to bend away after about 2 minutes. Electrostatic fields, humidity, mechanical cues, and temperature have all been ruled out. Some hypothesize that Phycomyces uses a volatile chemical signal that deflects around the obstacles with tiny air currents, but this is far from proven. Although Phycomyces is an unusually sensitive species, most fungi are able to detect and respond to light, its direction, intensity, or color, temperature, moisture, nutrients, toxins, and electrical fields. Like plants, fungi can see color across the spectrum using receptors sensitive to blue light and red light. Unlike plants, fungi also have opsins, the light-sensitive pigments present in the rods and cones of animal eyes. Hyphae can also sense the texture of surfaces, as one study reports that young hyphae of the bean rust fungus can detect grooves 0.5 millimeter deep in artificial surfaces, 3 times shallower than the gap between the laser tracks on a CD. When hyphae felt together to make mushrooms, they acquire an acute sensitivity to gravity, and as we've seen, fungi maintain countless channels of chemical communication with other organisms and with themselves. When they fuse or have sex, hyphae distinguish self from other and between different kinds of other. Fungal lives are lived in a flood of sensory information, and somehow hyphae, piloted by their tips, are able to integrate these many data streams and determine a suitable trajectory for growth. Humans, like most animals, use brains to integrate sensory data and decide the best course of action. Accordingly, we tend to look for particular places where integration might take place. We are like a where, but with different plants and fungi, asking where only gets us so far. There are different parts of a mycelial network or a plant, but they aren't unique. There are many of everything. How then do sensory data streams come together within a mycelial network? How do brainless organisms link perception with action? Plant scientists have wrestled with these questions for more than a century. In 1880, Charles Darwin and his son Francis published a book called The Power of Movement in Plants. In the final paragraph, they suggest that since root tips determine the trajectory for growth, it must be at the root tip that signals from different parts of the organism are integrated. Root tips, the Darwins write, act like one brain of one of the lower animals, receiving impressions from the sense organs and directing the several movements. The Darwins' conjecture has come to be known as the root-brain hypothesis and is controversial, to put it mildly. This is not because anyone disputes their observations. It is clear that root tips do direct the movement of roots, just as growing tips direct the movement of shoots above the ground. What divides plant scientists is the use of the word brain. For some, it is a proposition that can draw us towards a richer understanding of the plant life. For others, it is a preposterous suggestion that plants have anything even like a brain. In some sense, the word brain is a distraction. The Darwin's main point is that growing tips, which pilot roots and shoots, must be the place where information comes together to link perception and action and determine a suitable course for growth. The same applies for fungal hyphae. Hyphal tips are the part of the mycelium that grows, changes direction, branches, and fuses. They are the part of the mycelium that do the most. They are numerous. A given mycelial network might have anywhere between hundreds and billions of hyphal tips, all integrating and processing information on a massively parallel basis.